All right, hi everyone. This is the IGCSE Edexcel Human Biology past paper, and this is for November 2023. This is paper 2, as you can see over here. And for this paper, the total marks is 90, and it tests you on the entire syllabus, so all topics. All right, let's start off with question 1. All right, question 1. The box lists parts of the eye and parts of the ear, and then we have a list of words there, and the table has descriptions for parts of the eye and ear. Complete the table by matching. Okay, so I'm just going to highlight which parts are the eyes first. So this would belong to the eye. This is the eye. This is the eye, and so is this and this. And then the other ones are the ears. So this, and this. And we'll go through them one by one. Okay, let's go through the parts of the eye, starting with the retina, which we can see here. Your retina consists of light detecting receptors that are responsible for, absorbing light, and then that information will then be sent to the optic nerve, which you can see here. So the optic nerve is where sensory information is sent to the brain so that your brain can process what you're seeing. And next we have our cornea. So our cornea is basically allows for light to enter, and it kind of refracts the light a little bit. So it kind of bends the light a little bit so it can go into your eyes. And your pupil is the area in which light can go and enter the eye. And then lastly, we have the ciliary muscles, which are responsible for helping us focus on near and far objects, which we call accommodation. Let me just talk about that first. So here are the ciliary muscles. This is for focusing on near objects, focusing on far objects. Now, when you're focusing on a near object, right, the rays are kind of diverging. So when we're looking at it, you have to make sure that you bend the light so that it goes into your eyes and so that your eyes are able to focus on it. So in order to achieve that, we need a lens that is thicker. And in order to achieve that, we can see that the ciliary muscles, they need to contract, and that causes the suspensory ligaments to slacken or to become loose, and that's why the lenses bulge. And then on the other hand, if you want to focus on far objects, so if the object is far away, right, the wavelengths or the light is going to be a bit more parallel. So you don't need to bend the light that much. So you can see the ciliary muscles, they are relaxing, and that causes the suspensory ligaments to become tight. So let's write the stuff out for the eyes. Contain light receptors, that is retina. Controls the amount of light entering the eye, that is your pupil. Right? So if your pupil dilates, meaning that it gets larger, then more light can enter your eyes. Refracts light entering the eye, that would be your cornea. Okay. So first we're going to talk about the semicircular canals. This is responsible for helping us regulate our balance. And now we're going to talk about how our ears listen. So sound waves would enter through the auditory canal, and then it would cause these bones called the ossicles to vibrate. And when it vibrates, it amplifies this vibration and causes the cochlea, which you can see here. It passes on the vibrations to the cochlea, and your cochlea would contain receptors that detect that, and the information will then get carried from your ear to your brain, and then your brain would process that information. All right, so let's go over the table. So vibrates when receives sound waves, that would be the eardrum. Helps the body balance, that would be the semicircular canals. Transfers that vibration across the middle ear, that's the three bones, right? The ossicles. And then lastly, it turns vibrations into nerve impulses. That's your cochlea. All right, let's move on to part B of the question. So it says, explain why a person using both eyes find it easier to catch a ball than a person who is blind in one eye. So let me include a diagram here. It's a little bit blurry, but you can see that if a person has vision in both eyes, right? So each eye would give a different image. And so the parts in which they would overlap allow the person to have a better depth perception, meaning that they're able to tell how far away something is. And that kind of vision is called 3D vision, or also known as stereoscopic vision. All right, so we can say that each eye gives a different image or view, and when the field of view of the two eyes, let me just overlap, that gives better depth perception. This is called stereoscopic vision. All right, let's go over the marking scheme. So each eye gives a different image or different view. And the second mark is that gives better depth perception or allows people to judge the distance better. And that is what we call stereoscopic vision. All right, let's move on to question 2. Draw a label diagram of the cheek cell from inside a mouth as seen under a light microscope. So I've just included a diagram here just to show you what it looks like. So this is an animal cell, and you can see it's irregular in size. You can see the nucleus and also the cell membrane as well. So let's draw that out. Okay, we've got our irregular cell. Okay, and then we've got a nucleus, and then we just got to label it. So this would be nucleus, this would be cell membrane, this would be cytoplasm, which is the fluid that's inside, and those are your 4 marks. So 1 mark is for the irregular size, and then 3 marks are for the 3 different parts that I've just labeled there. Part B1, explain what is meant by the term tissue. So this is one of the definitions that you do need to remember. So it is a group of cells that are similar to each other and work together to serve the same or serve a particular function. All right, let's go over the marking scheme here. So we've got a group of cells working together. That is 1 mark. The second mark is similar to each other, and to serve a particular function. That's a 3rd mark. Part 2, state the type of tissue that contains cheek cells. So the type of tissue would be called squamous epithelium. And squamous just means that they're very thin. All right, part 3, name 2 types of other tissues. So you can say ciliated epithelium cell, which is what I'm going to write here. That's the ones that are found in your trachea. So they've got cilia, and they're used to push up the mucus so you can cough it out. So I'm going to write ciliated epithelium. And another one you can also say is your nervous tissue. So there's lots of answers that you can say for this. You can say nervous tissue, blood tissue, bone tissue, muscle, connective tissue, skin tissue. All right, let's move on to the next question. All right, question 3. A vaccine is used to protect people against a disease. The graph shows the level of antibodies in the blood of a child given the vaccine. Okay, let me just go over what vaccines are or how vaccines work. So I've taken this from the textbook. All right, so bacteria, pathogens, all viruses, all these things, they have something called an antigen, and you can see that there. So an antigen is what our body recognizes as foreign. And you can see different pathogens have different antigens. And so when we're using a vaccine, we are injecting a person with that antigen. So usually in the form of like a weakened virus, for example, or a dead pathogen, for example, or a dead bacteria, for example. So once these antigens are in our body, we have our lymphocytes here, which you can, let me just highlight this. We have our lymphocytes here that produce what we call antibodies. You can see that over here. Now, these antibodies bind specifically to that antigen. So, for example, this antibody here will bind to this pathogen, and this antibody here would bind to this pathogen. So it's very specific. Now, the reason why vaccines are effective is because you have what we call memory cells. So memory cells stay in circulation in our body so that when there is a pathogen, the same pathogen comes again into our body, the rate at which we produce antibodies is much, much, much faster. So the response is much faster, and that prevents us from feeling the full symptoms of that disease. All right, let's continue with part A. State the general name of given to disease-causing organisms. That would be pathogen. So, pathogen. All right, B1. Determine the time in years between the first dose and the third dose of the vaccine. Okay, first dose and the third dose. So we're looking at here versus here. Now, we can see this is 0.8, and this is 2 years, so 0.8 minus 2 minus 0.8 gives us 1.2. So 1.2 years. State the type of immunity that vaccination produces. Active artificial immunity. So what that means is, active means that our body is producing antibodies and it's making memory cells. And then artificial means that we're not being infected naturally, but we are injecting something that we've produced. All right, the next part, name 2 components that could be in a vaccine that would help to trigger immunity. So like I said earlier, it has to have something with the antigen to trigger the immune response. So it can be a weakened pathogen or a dead pathogen, or you could also use toxins as well. So that's indicative of that pathogen being there in that person's body. They even accept antigens as a part of the marking scheme. So I'm going to write weakened pathogen and antibody. Oh, sorry. Whoops. Antigens. My bad. All right. Part C. There are some antibodies in the blood of the child before the first vaccination is given. Explain why there are antibodies in the blood before vaccination. Okay. So the reason why is the child can receive what we call passive natural immunity. So passive because the baby is not producing their own antibodies, and natural because, this happens naturally. There are 2 ways in which this can happen. Both of them is via the mother, either through the placenta or across the placenta when the baby is still in the uterus, or through breast milk. This is because antibodies can be transferred from the mother to the child via the placenta or through drinking breast milk. All right, let's go over the marks. The first mark is transferred from the mother or passed from the mother through placenta or breast milk. That's the 3 marks. Part D, explain why a booster dose is necessary. So let's scroll back up to this graph that we see here. Right? We can see that once you have the first dose, the second dose, the third dose, we can see the level of antibodies in the blood drops pretty drastically, and that's why a booster dose is needed, so that the level of antibodies is above the unsafe level for antibody. We want to make sure it's above the line so that the person is less likely to get the disease. All right, let's answer part D. After the third dose, the level of antibody in the blood decreases below the unsafe level. This would increase the chance of the person catching the disease. Therefore, a booster dose is needed to increase the antibody levels above the unsafe level, so that the person would be more immune to the disease. All right, let's go over the marks. The first one, antibody levels would fall below the unsafe level. That's 2 marks over there. The third mark is greater chance of that person catching the disease. Fourth chance is the booster dosage is needed to increase the antibody levels above the unsafe level. And the fifth mark is so that the person would be more immune. All right, let's move on to part E. Give 2 differences between the effects of the first 3 doses and the effect of the booster dose. All right, let's go back to the graph here. Okay, so we're comparing the first 3 doses and the booster. One difference is that the booster dose causes a much greater antibody level, right? And number 2 is that the rate at which it drastically increases is much faster compared to the first 3 doses, right? So you can see it happens within 0.4 years, right? And the last difference is that you can see that the antibody levels remain pretty high throughout the years, right? So the rate at which it decreases is lower compared to after the first 3 doses. Okay, let's go back to part E. So we can say after the booster dose, the antibody levels are higher. And the other one is the rate at which the antibody levels decrease is slower after the booster dose. All right, let's move on to question 4. Read the passage below, use information in the passage and your own knowledge to answer the questions that follows. Okay, so I'm not going to read the whole thing, but I will highlight some points. So here we have statins. They are used to regulate lipid levels in the blood, and it's given to those who have failed to lower their cholesterol levels through diet and lifestyle, and it's used to reduce the risk of heart disease. So people with liver disease or kidney problems should avoid taking it, and also women who are pregnant and breastfeeding should also avoid. All right, in the UK, approximately 7.5 million people take statins. It's 12% of the population. However, up to 20% of these people have stopped taking it because of side effects, like muscle aches, feeling sick, and joint pains. So now they're doing an experiment where they're splitting people into 2 groups, people that take statin for 4 months and people who take placebo for 4 months, right? And then they rated how bad their symptoms were on a scale of 0 to 100, and this is the table. So part 8 says describe the changes in diet and lifestyle that could lower cholesterol. So in terms of diet, a person could reduce their cholesterol intake in their diet and could increase the amount of exercise they do. All right, that would be the 2 marks. Let's move on to part B1. Explain why people with liver disease or kidney problems should avoid taking statins. So we're going to split this question into 2, addressing the liver disease first and then addressing kidney problems. So one of the main roles of our liver is to break down toxins and drugs, right? And if a person has a liver disease, then they may not be able to break down statin, and that could accumulate in their circulatory system, in their blood. So I'm going to write that down. The liver is responsible for breaking down toxins and drugs. Therefore, if a person has liver disease, they cannot break down statins. All right, so the next part, what is the function of the kidneys? So the kidneys are responsible for excreting waste products. So whatever toxins are being broken down by the liver, that eventually gets excreted via our kidneys and then through our urine. So we can say the kidneys are responsible for excreting waste products such as the products produced in the breakdown of drugs. If a person has a kidney problem, then they would not be able to remove the waste products. Okay, let's go through the marks. The first mark is liver is responsible for breaking down toxins, therefore they cannot break down statins. The third mark is kidneys are responsible for excreting waste products. And the fourth mark would be, therefore, a person would not be able to remove the waste products. All right, let's move on to part 2. Explain why women who are pregnant or breastfeeding should avoid taking statins. So this is the case with pretty much all drugs, including alcohol, for example. So the substances can go from the mother to the baby, either across the placenta when the baby is still in the uterus, or it can end up being in the breast milk as well. They should avoid taking statins as it can be passed from the mother to the fetus via the placenta, and it could be present in the mother's breast milk. You could also add an additional point and say that that could affect the development of the fetus. So the first mark is statins can be crossed from the mother to the fetus by the placenta. That's actually 2 marks. And the third mark would be in the mother's breast milk. All right, part C1. Using the results of the study, calculate how many times greater the score for symptoms when taking statins is compared to the score when not taking tablets. So with versus without. So that's the placebo. Oops. So with it's 16.3, and then no tablets is 8. So 16.3 and 8. So let's simplify this first by dividing by the smallest number. We get 1 here, and then we get 2.0375. So it'll basically be 2 times greater. Or you can say 2.04 times greater. That's fine too. All right, part 2. Discuss the results of the study using information from the table. All right, let's scroll up to the table here. All right, so if we look at the statins and placebo compared to the tablet, we can see that the mean score is twice as much for both statins and placebos. It's around twice as much. But if we compare these two only, we can see that these scores between the statins and the placebo are quite similar. So this means that the symptoms might not be because of the statin, but because of something else. For example, their lifestyle or their diet or what they're eating. Okay, let's scroll back to part 2. When comparing the mean scores of the group without tablets versus with statins, we can see that the mean score is twice as high with statins. However, when comparing between statins and the group with the placebo, the mean scores are quite similar. This would indicate that the symptoms the group is experiencing may not be due to statins, but perhaps due to other factors like their diet or lifestyle. Okay, let's go through the marks. The first mark is comparing those with symptoms versus no tablets. We can see that the mean score is twice. That's 1 mark. The second mark is when comparing statins and the placebo, the mean scores are quite similar. That's a 2nd mark. The third mark is that symptoms that the group may experience might be due to something else, for example, their diet or their lifestyle. So that is the 4th mark. One more point that you can add is you can say that the symptoms are still present even when taking no statins. All right, so this is the first 4 questions done. If you want to look at the next part of this paper, just click on the description box below where you see the link of the next video. All right, bye.